Assalamualaikum and hello everyone. This is our video assignment. We are from class 17 and I hope you all enjoy and understand watching our video. So this is our group members, which are the first presenter is me Putri Diana, followed by Nur Fatiha, the next one is Nurul Hana, and lastly Nur Azli. Our journal title is the analysis of vitexin in aqueous extract and commercial products of Andean Passiflora species by UHPLC-DAD. These are the authors, the year of publication, and the publisher written in this slide. Next slide, I will explain the introduction. For significance, this plant have important economic, ornamental, and biological use. Some of the enopharmacological use and reported activities include diuretic, analgesic, anxiolytic, anti-inflammatory, hypoglycemic, antioxidant, antispasmodic, and neuroprotective activities. They are also recognized worldwide for their ethnopharmacological use, and different pharmacopoeias indicate vitexin as the chemical maker. In this context, the objective of this journal was to determine the vitexin content of some Colombian Andean Passiflora species and commercial products by ultra-high performance liquid chromatography. And the second one is to evaluate the usefulness of this flavonoid as a chemical marker of the analyzed species. Next, I will explain the meaning of this method validation. For linearity, to obtain test results that are directly proportional to the concentration of analyte in the sample. Next is sensitivity, which is the lowest analyte concentration that can be measured with acceptable accuracy and precision. The next one is precision, to evaluate repeatability and intermediate precision. The last one is accuracy, to express as the recovery percentage obtained after spiking a sample with known amounts of the standard solution.
Next, I will continue this presentation and talk about the methods in this study. This UHPLC analysis consists of multiple methods. Starting from the mobile phase, it uses the in-step mobile phase, and its illusion is the gradient illusion. In this analysis, the gases that was used are ultrasonic bath, and the pump then was used is the Dionex Ultimate 3000 RS with a detector that is diode array detection, that is DAD. The detection was performed at a wavelength of 340 nanometer, which is with its spectra at 200 to 450 nanometer. Next, the injector that was used with the amount of 3 microliter. Next, the flow rate is 0.3 milliliter per minute, with the columns used is within the temperature of 35 Celsius, with the height of the column, which is 2.1 millimeter. Its diameter is 2.1 milliliter and its length is 100. Not only that, 7 different concentrations was used for the calibration curve, which is 1.5, 6.0, 12.0, 24.0, 48.0, 96.0, and lastly is 144.0 microgram per milliliter. Lastly, it was filtered using a 0.22 micrometer with a mini-Q system. Next, I will be talking about the method validation that was used in this study, starting from the specificity. The specificity was based on the formula that was shown above. Next, I'll be talking about the accuracy. The accuracy was based on the drug product, which is vitexin. Not only that, the precision, which is based on the repeatability and intermediate precision. And lastly is the linearity, which is 1.5 to 144 microgram per milliliter. Lastly, I'll be talking about the method validation consists of limit of detection and also limit of quantitation. The limits of detection and quantitation in this study were determined by successive dilutions of calibration curve and also signal to noise ratio of 10 to 1. The relative standard deviation, which is the RSD, of below 5% was observed for LOQ, and the ratio 3 to 1 was observed for LOD.
Hello, my name is Nurul Hana binti Mazlan, and right now I will proceed with result and discussion. As described earlier, flavonoids are the most frequently metabolites reported in species of the Passiflora genus. Among the species analyzed in this study, all of these 7 species belong to the Tacsonia subgenus, show the most complex flavonoid profile, with several peaks corresponding to these metabolites. Next, the complexity of the extract composition was one of the challenges to be overcome when developed the analytical method. Although the developed methodology does not allow the separation of all flavonoid peaks, especially the complex sample, it still enables to differentiate the flavonoid fingerprints from each extract. Validation of analytical procedures was performed according to the ICH guidelines. The validated parameters were linearity and sensitivity, such as regression coefficient, LOD and LOQ. Next, position was evaluated as repeatability and intermediate position. Then, accuracy was expressed as the recovery percentage obtained after spiking a sample with known amounts of standard solution. Firstly, the value of regression coefficient obtained is 0.9991. From the regression coefficient obtained, the method developed for the quantification of the vitexin show good correlation between the response and the concentration of the flavonoid. The limit of detection and quantification value is 0.4 and 0.6 microgram per mil respectively. The next part is position and accuracy. 3 concentration levels of the standard solution were analyzed in triplicate within 1 day for repeatability and on 3 consecutive days for intermediate position. It can be concluded that all the parameters obtained in this table 3 were satisfactory to the theory since the RSD were found to be below 5% according to the limit recommended in the ICH guidelines. For the accuracy value, the reported data in this table 4 represent the average percentage of triplicate and its RSD. The mean value and the RSD value for the recovery percentage were 99% and 2.94% respectively. From the value obtained, the mean is very accurate and the RSD value was satisfactory to the theory since the value obtained was below than 5%. Next, I will explain about the vitexin quantification in the extract study. Figure 2 shows the crude extract of the species with quantifiable amounts of vitexin. It can be concluded that only 3 species obtain vitexin above the quantifiable limits of the method. From all these 3 species, Passiflora mixta shown the highest amount of vitexin extract. However, no vitexin was detected in these 4 species. Moreover, our method was able to detect vitexin in some species even at low concentration. For example, all of these 4 species shown a low, unquantifiable or undetectable concentration. Therefore, the poor presence of vitexins indicate that the low availability of this flavonoid as a chemical marker. On the other hand, Two vitexin derivatives that shown here were found as a commercial standard, could be used as preferable analytical markers and potential therapeutic markers, as they are the major compound in some Passiflora species.
Okay, now I will continue by making a conclusion to the topic of this presentation. Based on the result obtained in the validation parameters such as the precision, accuracy, LOD, LOQ, and so on that have stated in the result and discussion section, only 3 out of 10 evaluated in different Passiflora species contain quantifiable amounts of flavonoid, which is the vitexin content that being analyzed in this study. These three names are the species that have the quantifiable amount or the limit of quantification LOD more than 5% of flavonoids. Then, these 4 species did not contain flavonoid and the remaining 3 species contain below the LOQ. So, based on the result, the use of vitexin as a chemical marker for the quality control of highly cultivated Passiflora species in Colombia is not recommended. In this paper also, they have made a recommendation that this method, which is UHPLC-DAD method, also useful in the analysis of Passiflora product commercialized in Bogota. This is because this method possible to identify product with significant quantities, low amounts, and undetectable quantities of vitexin in that species of Passiflora. Okay, that's all from us. Thank you for listening.